Hello everyone! Today we are going to introduce single nucleotide polymorphism or SNP detection. SNP detection is an analysis method used to determine the presence and types of single nucleotide polymorphisms in an individual's genome. Through SNP detection, we can identify nucleotide variations at specific gene loci in individuals, allowing us to understand the genetic differences among individuals. This is of great significance in genetic research, assessment of individual health traits, prediction of disease risks, and personalized drug treatments, among other areas. Now, let's start by addressing a question. What is a single nucleotide polymorphism? SNP refers to genetic variations at single nucleotide positions in the genome. It is the most common form of genetic variation, with frequencies exceeding 1% in the human population. These variations can include single base substitutions or insertions or deletions, as well as other types of nucleotide changes. SNPs exhibit differences among individuals and can impact their genetic information and phenotypic characteristics, hence the need for detection. This video will provide you with a detailed explanation of SNP detection methods. The working principle of SNP detection can vary depending on specific detection method used. In today's video, I would like to illustrate how the detection process works using the example of a patient with a family history of gastric cancer who wants to determine his susceptibility of the disease. Step 1 is sample collection. DNA is extracted from patient's bodily fluids, blood, saliva, or other samples. We have discussed in detail how to extract DNA from samples in a previous video, and we will also provide professional extraction kits. Please feel free to contact us for more information. Step 2 is SNP selection. After DNA extraction, the target SNP to be detected are determined based on the specific requirements. Taking gastric cancer as an example, the SNPs of interest may include PTEN, CDH1, MUC1, among others. Step 3 is DNA amplification. The DNA segment containing the target SNP is amplified using methods such as PCR. This step generates a large amount of replicated DNA for subsequent analysis. Step 4 is SNP detection. With a substantial amount of replicated DNA, including the target SNP, the detection process becomes straightforward. In fact, we have different technological methods to choose from to achieve our goals, including A. Sequencing methods, using Sanger sequencing or next-generation sequencing techniques such as Illumina sequencing to sequence the amplified DNA fragments, followed by analyzing the sequenced DNA to determine the nucleotide variations of the SNP. B. Probe-based methods, utilizing DNA probes such as primer probes or probe arrays to hybridize with the target SNP region. The presence or absence of the SNP is determined by detecting specific probe DNA pairing. C is enzyme-based methods, employing specific restriction enzymes to digest the amplified DNA fragments and determining the nucleotide variations of the SNP based on the digestion patterns. D is primer extension methods, using specific primers to amplify the target SNP region and determining the presence or absence of the SNP based on primer pairing. E is mass spectrometry-based methods, utilizing mass spectrometry techniques such as genotype analysis using tandem mass spectrometry to detect SNP variations. In the case of our gastric cancer risk detection, all of the above-mentioned methods can be applied. The choice depends on experimental conditions. Step 5 is data analysis, analyzing and interpreting the obtained data to determine the genotypes of the SNP in each sample. Step 6 is result interpretation, evaluating an individual's genotype at the target SNP site based on the detection results, assessing disease risks, predicting drug responses, etc. For example, if mutations are detected in the PTEN, CDH1, and MUC1 gene loci, it indicates an increased risk of gastric cancer for the patient. In fact, besides the gastric cancer risk assessment we have been discussing, SNP detection has various applications in clinical settings. For example, it is used for disease risk assessment, particularly in the screening of genetic diseases and cancer. It can also be employed for developing personalized drug treatment plans and determining genetic relationships. These are just some common applications of SNP detection in clinical practice. With advancements in science and technology, the application of SNP detection in clinical practice will continue to expand and deepen. We will provide detailed explanations on the applications of SNP detection in future videos, so stay tuned. Why is SNP as a type of genetic variation worth specifically detecting? This is because SNPs are the most common type of genetic variation, which makes them easy to detect, identify, and track. Additionally, many SNPs are associated with specific phenotypic traits, making SNP detection highly meaningful. SNP arrays and sequencing technologies enable the detection of SNPs at thousands to millions of specific locations across the genome. This high density and genome-wide coverage provides comprehensive information about an individual's genetic makeup. In practical applications, SNP detection can be performed using non-invasive samples, making it more convenient and non-intrusive. It is also cost-effective and can be integrated with other genetic data to gain a comprehensive understanding of genetic variation and their functional significance. That concludes today's video. Thank you for watching.